Hello everyone and welcome to the chapter 11 movie. We're talking about the nervous system now, and this beautiful picture of a neuron. So the nervous system is very complex, and I'll try to make it as simple as possible. There's two basic divisions of the nervous system: the central nervous system, which consists of the brain and spinal cord. As you can see here, there's the brain and spinal cord. And its main function is information processing. Then we also have the peripheral nervous system, which is everything outside the central nervous system. And you can see it's divided into a motor and a sensory division. The sensory division consists of our sensory receptors, and it's going to send information to the CNS. And then the motor division receives information from the CNS and then causes some sort of action. We can see the motor division here is broken down into somatic, which is our voluntary muscles, and then our autonomic, which is involuntary, and we'll talk about it all about it in chapter 14. Now we need to know the three basic functions of the nervous system, and these are input, processing, and output. And we'll see different divisions of the nervous system do this, and we'll also see that different parts of the neuron, which is the basic unit of the nervous system, do this. So input, that's like your keyboard or your mouse, if we're comparing this to a computer. Then the processing, that would be the processor in the middle of your computer. That's where your thinking happens in your body. That's done in your brain. And then output, that's like the monitor or the printer. So that's the computer analogy of the three basic functions. Next, we're going to talk about neurons. The structural and functional unit of the nervous system is the neuron. And here is a typical neuron. And the three basic parts are the dendrites, which would be the input part, the cell body, which does the integration, and then the axon, which would be the output region of the neuron. So these things out here are called dendrites. These are the highly branched processes that receive input from other neurons. They sort of look like trees with all their branches. Then we have the cell body. This contains most of the organelles, and this is where integration happens. Neurotransmitters are chemical messengers. They're produced in the cell body. We'll talk about them later. The neurofibrils or neurofilaments. These are the intermediate filaments in a neuron. They provide structure, and they're used to transport things up down this long axon. And then we have the axon. Here's the axon, which is the output region. The axon hillock is this region right in here. It's the base of the axon, and that's where the action potentials are first generated. The axon lemma is just the plasma membrane surrounding the axon. And the axoplasm is the cytoplasm and the organelles in the axon. There's not many organelles there, but there's cytoplasm. Axons usually branch into what are called teledendria, and that way this neuron can contact more than one neuron later on. These end at what are called synaptic terminals, or axon terminals is another term for them. And each synaptic terminal forms a synapse between the presynaptic, so in this case the blue neuron here would be the presynaptic neuron, and the purple one would be the postsynaptic neuron. And at the synapse, here's a close-up on a synapse, so you can see there is a space there. It looks like they're touching, but there actually is a microscopic space. The cells communicate using neurotransmitters. That's the chemical messengers. That's these green things are called neurotransmitters. And the neurotransmitters are packaged into synaptic vesicles. The neurons send messages to other neurons, muscles, or glands. And if we look at these neurons, many of these have what are called collateral branches. These collateral branches allow neurons to communicate with different cells. So this neuron could be neuron 1 can communicate with neuron 2, and then this branch might go off on a third of neuron 3, and this branch might go through to neuron 4. One observation about neurons: most central nervous system neurons lack centrioles, and therefore they cannot divide. Now this is a good thing because once our brain is formed, we don't really want it to change. Forget everything we've learned. But unfortunately, this is why if your spinal cord is damaged, we can't repair those neurons. Hopefully, one day scientists are looking at it. So that's the neuron. Also, because they don't divide, most neurons live a very long time. They live until really your whole life. Alright, so we have some review questions. The nervous system has 3 overlapping functions, which is the logical order in which they occur. The first of that is sensory input and integration. So while studying for an exam, you reach for a beverage. To extend your arm, your somatic nervous system. What part of a neuron is the output region? That is the axon. So it's an axon because it's the axon. The portion of an axon that contacts its target is the axon terminal. Where are neurotransmitters made? In the complex or the cell body. And what part of a neuron is the input region? And the contents are maybe dendrites. What type of cells transmit action potentials? And the correct answer is neurons. We'll talk about these other cells in a minute. Neuron classification, we can classify them based on their structure or their function. So first let's look at structural classifications. This here is called an anaxonic neuron because it doesn't have an axon. These are found in the central nervous system, and their main function is information processing and integration. Bipolar neuron, it has one dendrite and one axon, and these are used for relay information the eyes. And then this one here, where the cell body is off to the side, this is called a unipolar neuron. And you can see the dendrite and the axon are continuous. These would also be sensory neurons, integrate or process any information and just relay information to the central nervous system. And this is the most common type, a multipolar neuron. We've got 2 or more dendrites and 1 axon. This is what our motor neurons look like. And then we can also classify them functionally as sensory, so those would be sending information to the central nervous system, and that would be like our bipolar or unipolar neurons. So that's the input part. We have interneurons, and those would be involved in integration. That's like a computer chip. And then we have the motor neurons, they're the output part. This picture that shows the relationship between the different divisions of the nervous system. So you've got sensory neurons sending information to the central nervous system, and then that's where integration happens, and then they're going to send the information out to the motor division, somatic or automotor. We'll talk more about that later. Alright, next we're going to talk about glial cells, or they're also called neuroglia. Their job is to support neurons. In the CNS, we have four basic types. Right here, these red ones are called appendable cells. They produce cerebrospinal fluid, or CSF for short. The CSF circulates, it produces and circulates the cerebrospinal fluid, which cushions and surrounds our brain and spinal cord. There are some special cells called epipendocytes, which help circulate the cerebrospinal fluid. And then there are tenocytes, which they transport substances between the cerebrospinal fluid and the brain. Then we have these here. This is a microglia. Microglia, they're kind of the immune system of the central nervous system. Our body's normal immune system doesn't reach the central nervous system because of what we talk about next, which is called the blood-brain barrier. The next type are astrocytes. So all these you see out here are astrocytes. They form the blood-brain barrier. As you can see here, they're surrounding this blood capillary. So anything that wants to get into our brain has to go through this barrier first, and it can keep some drugs out. Some drugs go right through it. These astrocytes, they also regulate ion, nutrient, and gas concentrations around neurons. They can absorb and recycle neurotransmitters. They form scar tissue. So if you're not sure, guess astrocytes. Also in the central nervous system, we have these neurons down here called oligodendrocytes. They form the myelin sheath, which you can see right here. The myelin sheath, it slides axons and it speeds up action potentials. Myelinated axons appear white, and they're called white matter. So here we can see white matter. And then unmyelinated axons. Unmyelinated axons along with their directed cell bodies appear gray, and they're called gray matter. So in the PNS, we have Schwann cells and satellite cells. Schwann cell, this is showing how important is the damage. See some satellite cells, they surround the cell bodies and regulate the environment like the astrocytes and the CNS. Another important function of Schwann cells is they help when a neuron is damaged. This figure shows that. So step 1, there's damage, the axon degenerates, the myelin degenerates as well. Step 2, Schwann cells are able to grow much faster and easier than neurons, so Schwann cells start to grow along this path, and macrophages can compete with the debris. Step 3, the axon starts to regrow along the path created by these Schwann cells. And then finally, step 4, the Schwann cells will surround this Schwann axon. Alright, here for some more review questions. What type of cells circulate CSF? And the answer is appendable cells. What type of cells ache for neurons to their nutrient supplies? Maybe the answer is astrocytes. What part of the neuron is the output region? Maybe the correct answer is axon. Alright, next we're going to talk about neurophysiology. So there's a couple different things that happen in order for cells to communicate. So first we've got to talk about the resting potential or the transmembrane potential. Then we talk about gradient potentials, the action potentials, and then we talk about what happens with the synapse and the primary information processing. So we have what's called the transmembrane potential, which is the difference in charge across the cell membrane. So here's inside of the cell, here's the plasma membrane, here's outside of it. So it's negative inside. This is due to differences in ionic concentrations and negatively charged proteins inside the cell. Cells are like bananas in salt. So they have high sodium outside. Salt is sodium chloride, so there's lots of sodium in this water. And bananas have a more high potassium, so there's lots of potassium inside the cell. The resting potential, we're going to watch this resting potential movie. Finish this. We're going to go to the master EMP site. Go to AMPFlex and watch the resting membrane potential.
About 100 billion nerve cells, called neurons. A neuron, like every other cell, has positively and negatively charged ions, inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of minus 70 millivolts. The resting membrane potential depends on two factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at minus 90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to minus 70 millivolts. Sodium-potassium pumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential.
So now we're going to explain this. The resting potential is due to weak channels, which are always open. An undisturbed cell has potassium leaking out, sodium leaking in, and the sodium-potassium pump working. And ions move across the membrane because of their electrochemical gradient. The electrical gradient is due to the fact that opposite charges attract and like charges repel. And then the chemical gradient is just diffusion. Things always move from high concentration to low concentration. And the equilibrium potential is where the chemical and the electrical forces are equal and opposite. Now I'm going to ask some questions. Value of the resting membrane potential is -70. Concentration gradients exist across the plasma membrane. Specifically, there are, and we have more potassium ions inside and more sodium ions outside the plasma membrane. So we read this correctly. Alright, the plasma membrane is more permeable to, the correct answer is potassium, because there's many more potassium leak channels. And the sodium-potassium pump transports, and it transports sodium out and potassium into the cell. That's how it maintains that concentration rate. The resting membrane potential depends on each atom below except, and concentration value for sodium ion is necessary. The greater permeability of the plasma membrane of potassium ions is necessary. The greater number of potassium leak channels, that's why it's more permeable to potassium. And C, the greater permeability of the membrane to anions rather than cations, that makes no difference. So C is does not depend on. Okay, so these are leak channels which are always open. And if you look at an undisturbed cell like this one, it will have potassium leaking out, there's a potassium channel, and it will have sodium leaking in. And then we've got the sodium-potassium pump working, sodium out and potassium in, to offset those leak channels. Okay. And again, ions want to move because of their electrochemical gradient. So let's move on now. This is not a real cell, a hypothetical cell where it was only permeable to potassium. So only potassium can get through this plasma membrane because we have just potassium channels. And if a cell was permeable only to potassium, we would have the electrical gradient because it's negatively charged inside the cell. That would be pulling potassium in, but because potassium's concentration is higher inside the cell, that would be pushing potassium out. And so the electrochemical gradient would be the sum of these, and in this case it would be out. And if we were to just let this cell go, potassium would leave until it reached its equilibrium potential, which is where the chemical and electrical forces are equal and opposite. So at this point there would be no net movement of ions. So at this point potassium wants to leave because of the concentration gradient, but it also wants to enter at the same rate because of the electrical gradient. The negative charge in here is pulling in these positively charged potassium ions, pulling them back into the cell. So that's what would happen if the cell was only permeable to potassium. Now if the cell was only permeable to sodium, the sodium concentration is in, because sodium concentration is higher outside the cell. And sodium is also positively charged, so the electrical gradient would be pulling it in. So its electrochemical gradient would be being pulled out. And if the cell was like this, the ion would continue to flow in until it reached a charge of plus 66. At that point, the chemical gradient is still going in, but now there's so many positive charges in the cell that starts repelling the positively charged sodium ions. So these would be equal about plus 66. Now, we know that a cell at rest is much more permeable to potassium, and that's why the resting potential is close to potassium's equilibrium potential. So we're at about -7 for the cell's resting potential. Alright, so now we review questions. The voltage displayed across the resting neuron membrane is termed the resting membrane potential. The resting plasma membrane is more permeable to, the answer is potassium. Maintenance of the resting membrane potential is due to sodium ions passing through the plasma membrane, actions of the sodium potassium bond. Sodium ions spontaneously crossing the interior of the neuron or IEs. And the correct answer is the sodium potassium pump, because it pumps them back against the concentration gradients and they sodium ions do not pass through the plasma membrane, they have to go through ion channels, and potassium ions do not spontaneously cross the interior of the neuron. Alright, the resting membrane potential depends on which atom below accepts. And the correct answer here, concentration gradient for sodium ion is needed. Rate of permeability of the plasma membrane to potassium ions, that's needed. Rate of permeability of the liquid anions, magnetic ions, that's not true, so that's the answer. The sodium potassium pump transports sodium out and potassium into the cell. And concentration gradients exist across the plasma membrane. Specifically, there is more potassium ions inside and more sodium ions outside the plasma membrane. And the value of the resting membrane potential is about -7. Alright, so next, that's the resting potential. Now we're going to talk about gradient potentials, but first we need to talk about ion channels. So here are some ion channels. Ion channels allow ions to cross the plasma membrane and change the transmembrane potential, because remember ions are charged particles. And what we're seeing right now are chemically gated channels. These open when a chemical messenger, or we call them neurotransmitters, neurotransmitter binds to them. So we can see when nothing is bound, they're closed. When a neurotransmitter binds, as we see here, the channel opens. And when no neurotransmitter is bound, the channel closes. And the next type of channels we're going to talk about are voltage-gated channels. And these voltage-gated channels, they open in response to changes in the voltage or charge. So at a charge of -70, this is closed. At a charge of -60, this opens. And then at +30, this inactivates. So this is a different picture of a voltage-gated channel that might help explain how it works. Remember that these channels are proteins, and proteins are made up of amino acids. And what we have here, this region of the protein has many positively charged amino acids. And remember that opposite charges attract. So when the interior of this cell is negative, it's attracting these positive charges. But if the voltage changes due to some positive ions entering the cell, those positive charges will repel these positive charges and open the ion channel. So that's how a voltage-gated channel works. And we also have some mechanical gated channels. They open in response to physical disturbance of the plasma membrane. This is how our touch receptors work, or our hearing receptors. We'll talk about them later. Alright, so now we're ready to talk about graded potentials. Graded potentials are localized changes in transmembrane potential. So if we look at a cell at rest, its chemically gated sodium channels are closed. That's what these are. These are chemically gated sodium channels. They're closed. Sodium wants to come in, but it can't go through the plasma membrane, and all the ion channels are closed. When a neurotransmitter binds to the chemically gated sodium channel, it opens and positive sodium ions enter the cell. So the cell went from -70 to -65 because positive charges enter it. And then these sodium ions would spread out and generate what's called a local current. So it's going to spread out in both directions. But as we see here, the change in charge, polarization is most, it's biggest here right where the stimulation happened. It changes all the way to -60, but as it spreads out, it's only a change to -65. And as it spreads out even further, it goes down to -70. This is just another way of showing that right where the stimulation happens, where this axon terminal reaches, that's the most change in transmembrane potential. And then as you get farther away from that site, it goes down until eventually it goes down. So the degree of depolarization decreases with the distance away from the stimulation site. Also, we'll see that change in the transmembrane potential depends on the size of the stimulus. A greater stimulus would lead to more ion channels opening. This would lead to more sodium ions entering and a greater change in charge. Now, when a chemical opens sodium channels, positive ions are going to enter the cell and the transmembrane potential becomes less negative. That's what we see here. The depolarization, or an EPSP, which stands for excitatory postsynaptic potential. When a chemical opens potassium channels, which is this one, potassium is going to leave the cell, so those positive ions leaving the cell makes the cell even more negative, and this is called a hyperpolarization, or an IPSP, which stands for inhibitory postsynaptic. So graded potentials, before we get to that, graded potentials are short-lived, they're local, they're passive, they don't require any energy, and they're variable in size depending on the strength of stimulation. So greater stimulation leads to a bigger graded potential. It could be 5 millivolts, it could be 10 millivolts, it could be 20 millivolts, depending on how big the stimulation is. So let's do a little review. Graded potentials, long-lasting, long-distance sensor self-generating. None of those are true. Graded potentials could be depolarizing or hyperpolarizing. That's true. Are not caused by the opening of voltage-gated channels. Are caused by the opening of chemical-gated channels, so A and C is correct. Alright, next we're going to talk about action potentials.